Thanks for coming to Neurology Grand Rounds. Many of us have been at the conference on retroviruses and opportunistic infections this week. And for that reason, we're very lucky to have Kevin Robertson come and talk to us. Kevin is a neuropsychologist whom we have known forever. I think I've known Kevin for 25 years as a colleague and friend. And anybody who works in the HIV, the neuro-HIV field knows him, not only because he's really fabulous in his field, but he's also incredibly collaborative. So all of us have tapped him to help us with one project or another. And he was probably the first of probably anybody in the neurology group who wanted to do international work, looking at neurocognitive disorders in HIV and the resource-limited setting. I think he's going to talk a little bit about that to us today. So, welcome, Dr. Robertson.
Thank you very much. That's a great introduction, Christina. I take it back. I couldn't have done better.
Tell me where you went to college or anything else.
You don't need to know. It's not important. Who cares after you've already done it? So thanks very much for having me and for being here. I'm going to talk about the background of HIV and then go through the changes to the definitions of HIV-associated neurocognitive disorders using the rule of three, which I'll explain, and then talk briefly about pathogenesis, which we don't quite understand completely, but a model, and then some data from UNC on compartmentalization, and then I'm going to talk about what I've done for the past dozen years with the help of Christina. Resource-limited setting work. Two studies in particular that address that, the International Neurological Study and the Neurocognitive Normative study that follows on to that and makes it comprehensible. And then talk a little bit about the future. So, this is the virus. This is what it looks like. This is the gene map. And HIV is split into two groups, 1 and 2, and we're really worried about 1 and specifically group M, but each one is a separate group. M stands for main, which is the most prevalent. N is new, non-O and non-M. O is an outlier, and P is a relatively new group that resulted from a single transmission from a gorilla. It is a virus that resulted from a zoonotic transmission from chimpanzee to human, and from this SIV specific virus to HIV-1M. The predecessor they've timed all the way back to 1625 or thereabouts, and the resulting virus that led to our current problems probably originated in the 20s and 30s, and subtype B, which is problematic in the resource-rich world, in the 70s. And the way it spread is kind of interesting. It came from the epicenter of the Democratic Republic of Congo, and then from there to Central Africa, and from there to East and South Africa, and from there to Haiti, and from UNESCO workers in Central Africa, which was then brought to the U.S. Through homosexual tourism to Haiti and the rest of the world. And here's just a look at genetic variation.
May I ask how they unraveled that sequence, that travel arc?
They did a lot of genetic sequencing and historical stored samples, and and a lot of complicated genetic analyses that are beyond the scope of this talk. But it's a really neat, fascinating history and story that has been well studied and well characterized. So this is sort of the differences in the virus, and I'm going to come back to this, but as you can see from this pie chart, most of the virus in the world is type C, which is the bottom white part of the pie. And what we deal with is clade B, which is the pinkish, or I guess red as it projects there, 10%. So HIV gets into the CNS within days, which brings us to neurology, and establishes a chronic, latent, low-level infection. And a few years ago, we went through, and Christina was part of the group that NIH charged to reformulate the diagnoses, and we came up with this set of this nomenclature: ANR, or asymptomatic neuropsychological impairment, mild neurocognitive disorder, and HIV-associated dementia, which I'm going to go through in a little bit more detail. But here's what it looks like, and the broad, overwhelming majority of people that do have HAND have the ANI variety, and it results in abnormality in only 2 cognitive areas on testing, and then there's increasing severity for the MND type of disorder with functional impairment, and then marked functional impairment and neurocognitive disorder with HAD, which is what we used to see in the old days. So here's the rule of 3, and it's just a way for me to keep all of this stuff in mind, because I like acronyms, and it's easy to clump it this way. So there are 3 criteria, and one is the severity, the neurocognitive impairment. Then there's functional impairment, whether or not it has an impact on activities of daily living. And then what confounding conditions there are that contribute to the diagnosis and cloud the picture or make it clear, on the other hand. So for the first one, there are 3 levels that I just went through. There's the mild level, asymptomatic neurocognitive impairment. There's the moderate level, mild neurocognitive disorder, and then there's the full-blown HIV-associated dementia. And then the second area in the rule of 3 is functional impairment, or activities of daily living. There's normal, mild functional impairment, and then marked. And then from confounding conditions, importantly, there are minimal or non-contributory things that maybe mild depression that doesn't have an impact on your daily functioning. There are contributing conditions such as substance abuse that isn't marked enough to impair your work ability. And then there are completely confounding conditions like CNS mass, severe substance abuse leaving you completely unimpaired, and you just can't make the diagnosis in that category. So here's the block diagram of all of this. You have to rule out pre-existing causes, make sure delirium's absent, and then we went through the cognitive ability areas that have to be impaired, and then the daily functioning. So what are the consequences of HAND? Well, there have been studies about what happens if you do have HIV-associated neurocognitive disorders, and as you might imagine, there's decreased adherence to medication, impaired driving ability. We've talked about decreased difficulty with ADLs, such as finances and meal preparation, and then you're 5 times more likely to complain of problems at work and twice as likely to be unemployed. And most importantly, probably, you have a shorter survival time. So those are the consequences. Here's, I'm briefly going to go through this complicated model of pathogenesis, or what we think might be happening. And what I'm going to show you is, in the middle here is a capillary lumen, and on both sides is parenchyma. And then you can see the different cell types here. And the red things, the things in red are that's virus from the blood. And then the things in blue are going to be virus from the CSF. So the important cells, we think, are macrophages and microglia. And we know that virus comes in it's transmitted in through T cells and monocytes and sets up, we think, a resident infection, which is shown in the blue viral particles, and then releases inflammatory markers that molecules that then impair neuronal function and end up killing neurons eventually. So another thing so that's the model as it stands. Inflammatory markers due to viral. The virus doesn't directly kill neurons. It's a bystander phenomenon. Ron Swanstrom at UNC has done a lot of work on compartmentalization and looking at virus within the CNS relative to the plasma. And we've collaborated with him through a number of his fellows, Kim Ratola, Pat Harrington, and Gretchen Snell. Over the years. And the virus in those that have HIV-associated dementia looks different within the CNS than it does within the plasma. I'm going to show you a little bit of those in just a second. And then typically those that are neurologically asymptomatic, there's an equilibrium between both compartments, both systemic and the CSF. So here's somebody that's neurologically asymptomatic, and there are two examples here. Let's see if I can get the mouse to work. But again, everything in red is blood and everything in blue is CSF. And what you can see here is that in this genetic tree, that there's pretty good intermixing of the genetic sequences here, and that there's not like one tree out here that's all red or all blue, and there's pretty much equilibration between blood and CSF in both of these subjects that were neurologically asymptomatic. Now,
Can I ask a question?
sure,
So those are genotypes, so that's all from one time collection
that is one time point.
at the same time?
I will, yeah, one time point with both plasma and CSF. So this is an example of somebody that has pretty much mixed, and they're asymptomatic neurologically. So, and here are examples of patients that have compartmentalization. And you can see that the blue circles highlighting out the trees that are, the branches that are compartmentalized in these subjects. And all of these subjects had HIV-associated dementia or had. And these boxes just outline the CSF that's unique. And then one or two intermixed. So this is, as you noted, those were cross-sectional. And this is a patient that was followed over time. This is somebody that was on study, and we had multiple samples. And while this is incredibly complicated, it is, if you look here, this is time 0, which was had diagnosis. So that's diagnosis of dementia. And the triangles are plasma, and the circles are CSF. So this is months from diagnosis. So this is 48 months from diagnosis of dementia, 41. Then at had diagnosis, and then 1 and 2 months after. And what you can see is that in the beginning, there's CSF over here and around, and over here later on, after the after diagnosis of dementia, and it just shows that sort of the evolution of this sequestered and compartmentalized virus that's unique and separate from the plasma compartment. So the CSF is set up sort of in what we think is an autonomous infection within the CNS and reflected in the CSF here. So that's a little bit about pathogenesis and what we know there. Now, as most of you know, prior to effective antiretroviral treatment in the mid-90s, up to 20% developed HIV-associated dementia. And then after that time, there was a dramatic decrease in HIV-associated dementia. However, those that are living longer there's an increased prevalence of the milder forms of neurocognitive impairment, which I'll show you. So this is just a slide to show you data from the multi-center AIDS cohort study, and that's a national study. And you can see in the mid-90s the dramatic decrease of all the CNS opportunistic infections, cryptococcal meningitis, toxoplasmosis, PML, and primary CNS lymphoma. And how when effective ALLRT was introduced, they all sort of, we don't see them anymore really. Unless somebody's really late to treatment and undiagnosed, you just don't see these CNS ARIs too much. And along with that, a dramatic decrease in HIV-associated dementia. So all well and good. But recent data from large cohort studies that Christina was a big part of here has found that there's still a fairly large prevalence of neurocognitive impairment out there in HIV-positive patients, even when you account for these confounding factors and you pick the cleanest group, those that don't have substance abuse, don't have other things that can account for it, you still see about 40% or so of neurocognitive impairment. So, and this just reiterates that, and this was a large study. Overall, it was 52% that had impairment, and if you looked at the cleanest group, it was about 40%. So although treatment is better, HIV-associated neurocognitive disorders are still prevalent, just in milder forms, but they still have an impact on daily function. And it's not really been addressed in resource-limited settings at all. Almost all of this research has been done in Europe, the US, and Australia. So why look at resource-limited settings? Because the burden of the virus is greater. By and large, of the 35 million people infected with HIV, most of it is in sub-Saharan Africa, as you can see here, or other resource-limited countries like Asia. And this just reiterates that. Those are people living with HIV, and these are people dying with HIV, and these are the most recent statistics from UNAIDS and WHO, and the bulk of the deaths are in sub-Saharan Africa. And then the new infections, of course, follow the same trend. They're all, or mostly in resource-limited settings, mostly sub-Saharan Africa. So about 6,000 new infections a day. Most of those are in sub-Saharan Africa, and most, and it's supposed to be on the women, I'm not sure what it's presenting there, but every minute a young woman is newly infected with HIV, and it's the leading cause of death of women of reproductive age, and they're twice as likely to become infected relative to young men. Another reason to look at resource-limited settings, as I alluded to earlier, the virus is different. And here's one pretty map of the virus and the different subtypes of group M. And you can see that it's different across the world. It's there are at least 10 distinct subtypes and then a bunch of circulating recombinant forms. Here's, you can see in the boxes all the different recombinant forms, so it's massively recombinant. C and AE are the most prevalent subtypes. AE is mostly in Thailand. They're responsible for the vast majority. C is mostly in sub-Saharan Africa. And relatively little is known about the relationships of those virus subtypes, their biological properties, and pathogenicity. And this is just a look at some of the recombinants. A and sub, you can see below in colors the subtype, what these pieces are made up of. So AE, which is a very common one in Thailand, is made up of the red and subtype A and the yellow and subtype E, for example. Now, this is a schematic that kind of gets at how different the virus is around the world. Each color represents a different continent or country, and except for the black. And the black is the Democratic Republic of Congo, sort of the epicenter of HIV-1. And you can see black all over this genetic tree. And then blue is the rest of Africa. And then you can see that North America or the Americas, et cetera, they're all sort of clustered down here in the B tree. So little genetic variation here, where there's wide genetic variation in the DRC and sub-Saharan Africa. As this just recapitulates, you can see in these pie charts that there are many different colors in these areas, and then when you get down to South Africa, it's all C. It's very uniform. So as I said before, most of what we've worked on has been done in neuroaids. It's been done in the developed world, and we knew that the prevalence was high prior to initiating treatment, but the prevalence was unknown in many resource-limited settings. And in 2003 or so, we had the idea to extend what we've been doing in the U.S. in terms of neuroaids and into sub-Saharan Africa. And I'm not sure, I think maybe Christina took this picture.
I took that picture in Malawi.
Yeah, it's in between the single neurologist in Malawi took us out to Mount something or another, and along the way there was this woman walking with her baby, and she posed for this picture. And that was beautiful countryside. So we decided that this was unknown and unstudied, and a way to start intervention would be to do a clinical trial in these resource-limited settings. And we set up the first international clinical trial in HIV. It was a collaboration between the AIDS Clinical Trials Group and the National Institutes of Mental Health. And it was a cross-NIH Institute collaboration where NIMH contributed funds to the National Institutes of Allergies and Infectious Disease to provide the infrastructure to get the study done. It was a lot of moving parts that ended up moving in the right direction slowly, but it did work. It was a multinational collaboration, and it took consultation with the big boys, as you can see here, And we ended up collecting 860 subjects. Our target was 80 per the 11 sites that we studied, so we got kind of close. They were all co-enrolled with a larger treatment trial, 5175, which was partnered with HPTN and part of the same study, HPTN 052, which made the cover of Science, made Mike Cohen a famous guy because it was the study that showed that treatment worked as prevention. We enrolled treatment-naive participants with CD4 counts under 300, and we gave them one of these 3 antiretroviral treatment regimens, which were World Health Organization recommended treatments at the time as a first-line regimen for resource-limited settings. And then this is outside of Queen Elizabeth's in Blantyre, just right across the road there. And that's where you went and got lunch. And this is Dick Price in the foreground, and from UCSF, who was one of our co-chairs on this study. And then Johnston Kamwenda is singled out with the arrow there. And the clinic was actually set up in this old leprosy clinic. You can see it's sort of shut down, but we were in the process of restructuring and setting it up as an AIDS clinic for doing this clinical trial. Here are the sites that we worked in. 2 sites in Brazil, Rio and Porto Alegre in the south. 2 sites in India, Chennai and Pune. 2 sites in Malawi, we just talked about Blantyre. And a long way, a site in Lima, and a site in Chiang Mai, a site in Harare, and 2 sites in South Africa, Durban and Johannesburg. Here's the hospital in the long way, and you can see that it's crowded. We developed, Christina was an intimate part of developing the neurologic exams, and we had these field tested, and we're told that they were incredibly long. We should cut them down after field testing, and which we did. We revised them, sent them back out there, and then developed a bunch of video training with Dr. Colin Hall and Dr. Price, along with Dr. Mora, and went out and trained the sites. And a brief neuropsychological exam. We had, I've lost count of how many different languages to deal with in these 11 sites, but it was amazing. We also had literacy and numeracy issues, so we chose to focus on a primarily motor-based battery, which included time gait, finger tapping, groove pegboard, and both of those tests we did dominant and non-dominant hands, and then verbal fluency. And because the agency didn't want to fund collection of normative data for these tests, which they said, well, just use US norms. Okay, which I'll get back to. We only looked at changes from baseline to follow-up with these exams. And here's Colin Hall doing an exam on a woman with severe neuropathy, and in the foreground here is Cecilia Kanyama, who is responsible for the Longway site, along with Nina Hasunapar from UNC, who lives in the Longway full-time. And this is another startup picture with Colin over here, and the head of the ACTG at that time, Connie Benson, and the Chiang Mai site, Neuro site PI, Kwanchas Brick in here, and this guy over here, who some of you may recognize from the Vaccine Trials Network. And this was a time, this was early, this was 2004, there was a lot of death, and everywhere you went. This is a picture from Harare, and this was not uncommon. There were coffins for sale everywhere and a lot of death. So when you went there, you felt like you were actually doing something. So what do we find? Overall, at baseline, we found a lot of country variation. We expected that. Differences in culture, perhaps HIV subtype differences, but what we found between the treatment arms was that they were equal. And if you put somebody on antiretrovirals, didn't matter which arm you put them on, they got better. Overall abnormality improved. The CNS dysfunction part of the neurologic exam improved. The peripheral part of the exam improved. And then all the neuropsych test improves across the arms. This just shows the disease markers. This is virus, and this is percent suppressed, the proportion of those who completely suppress the virus below the detectable levels, and these are the countries that were involved, and this is the subtype for each of the countries. But as you can see, they rapidly suppressed virus on study and pretty much remain suppressed. And this is CD4 counts and a measure of immune function. As you can see, as they went on, their CD4 counts improved. This is a look at one of the neuropsych tests, semantic verbal fluency, and just to demonstrate the wide variety and variation across the countries, which screams getting normative data. And this is the longitudinal data, also screams get normative data. And this is the gross motor task of time gait. But the long and the short is that they all improved across time. And here's the one of the tests by treatment arm, and you can see red, green, and black. The black arm was arm B, and the wide variation you see there is after week 48 or 72, they deemed that arm to be inferior and rolled everybody into other arms. So there were very few people that stayed on that arm, as you can see by the incisors in parentheses down below. So week 96, there were only 99, and out to week 192, there were only 32 people on that arm. But the other arms had a fair number of people all the way out to week 92 that stayed on treatment. This is probably doesn't project well, but is the overall look at what happened on the neurologic exam. Here is in yellow, which is the famous, the graph on suppressed in viral load, and this is the CD4 counter immune functioning. And then these are down here are decreasing markers off of the neurologic exam, neuropathy, all neurodiagnoses, and overall neuroassessment. And they all sort of decreased over time, and significantly so. So we got to the end of the study, and NIMH said, well, we want to know about impairment. Well, we couldn't really tell them about impairment because we had no context to put this in. So, and we also convinced them that it was necessary to support the infrastructure there in these resource-limited settings. There's no normative data. If they have normative data, they can use it both for neurological research as well as clinical studies longer term. And in fact, for 5199, the study I just presented, we had no way to determine what was normal and what was impaired. So we talked them into and got a grant to do the normative study, and we enrolled 2,400 HIV-negative subjects and stratified them by what we felt like was important and what we could actually do within 2,400 HIV-negative subjects. We expanded the neuropsychological battery to include verbal learning and memory, executive functioning as well as memory, and added a 6-month follow-up to look for practice effects in normative data. We also gave them a neurologic exam. And we used the same sites that we used in the parent treatment study, which are presented here on the map. They had to be what we would hope were high risk. There was no guarantee they were high risk, but they were presenting to voluntary counseling testing centers within 30 days of enrollment in our study, presumably because they felt like they were at risk for being infected. So we wanted them to be as much like our HIV positive patients as possible, and this was the way we felt like we could get to that point. We enrolled 240 participants at each of 10 sites and stratified by 2 educational levels, 10 years, over 10 years and under, and 2 age levels, over 35 years and under, and male and female, and created a normative database to apply the data to our treatment study. And this is what we found in our treatment study. By and large, in these resource-limited settings, we're seeing very much what we would see in a US or a European study in terms of neurocognitive impairment. So many of them are normal, and at 54%. There's mild impairment in 25%, and 17 had moderate impairment, and 3% had severe impairment, consistent with HIV-associated dementia. And while the study was started before the Frascati criteria, the revision of the diagnostic nomenclature that I went through, we did not have functional diagnoses, so we can't, with perfect ascertainment, say that they had MND or ANI or had, but we're close on the neurocognitive part of it. What this looked like over time, you can see at the bottom axis is baseline at 0, and then weeks out, 24, 48, 72, 96, et cetera, et cetera. And then on the percent neurocognitive impaired, you can see that what happened over time was that mild impairment went down somewhat, moderate impairment really went down, and severe impairment went down but didn't have much room to decrease. So it started at 3% and ended up close to 0. And this is just the overall improvement in the normals over time, and the percent of who was normal. And this is a tabular format of that same data. So it's the same thing, just not as pretty. So at baseline, 46% were impaired, and by week 92 on antiretroviral treatment, only 22% impaired. So what did we learn? We learned that neurocognitive impairment is prevalent in resource-limited settings, that you improve when you put people on treatment, and it didn't really matter which of these arms we put them on. There was a difference between the arms and how well these drugs got into the CNS, but in the grand scheme of things, it didn't matter as long as you were suppressing virus, and that was the important point. We still had performance that varied by country, and we're looking at subtyping those. We've genetically subtyped a subset of that sample, and we'll be correlating that with neurocognitive outcomes to see if there's a difference there. So why worry? We have antiretroviral treatments. We can treat severe HIV neurocognitive disorder. Well, we worry because there's still a lot of neurocognitive impairment in those who are virally suppressed. Christina's site here, along with Ann Collier, was part of a prospective study that was done a number of years ago that looked at neurocognitive impairment in those people who were antivirally suppressed for quite a while. And what we found was that there was still mild impairment in 39% of those individuals who were virally suppressed and healthy, and mild to moderate impairment in 26%. So it's still an ongoing issue, and prior to a recent study that came out, we really didn't know whether those individuals were going to progress or not in that asymptomatic neurocognitive category. But Christina's charter group was part of a larger study that looked at whether or not this would have clinical significance and lead to progression. It was a big question, and still is a big question in the field. A lot of infectious disease clinicians think that asymptomatic neurocognitive impairment is smoke and mirrors. It's just noise in the system and not really relevant. In fact, if we had to do it all over again, we would not name it asymptomatic neurocognitive impairment. We would name it subclinical or something else. Because when you say something's asymptomatic, people say, well, why are we looking at it? There's no symptoms. So, but this study in Charter had 33% of their 1,500 patients at entry who were ANI, and they looked at what happened to them over time. And what happened was, if you look at the normals versus the asymptomatic individuals, and they were fairly similar, except for a little difference in nadir CD4 and the ANI group on a bunch of demographic and systemic disease characteristics. What happened in this graph is, and in the blue are the normal, neuropsychologically normal, and in the red line is the asymptomatic neurocognitive impairment. And if they self-reported functional impairment, they were much more likely to become mild neurocognitive disorder. So they progressed. And this was, to me, this was something that answered a really relevant clinical question. This was presented earlier, but it's just recently been published, and to me, it's like a really one of the most important papers in the last year or two. Now, some people said, well, this was self-report data on functional impairment. People complain all the time. Who cares whether they're complaining? Everybody complains. You're more likely to be depressed. If you're depressed, you're going to complain. If you're anxious, you're going to complain. Well, they also did performance-based functional impairment, real performance tasks to look at this. And they found an even higher risk of progression in that case. So it's real, and there is clinical significance to ANI. So here are the statistical values, but you can see it on the graph much easier. So I've said all of this. It leads to an earlier decline, and there's an increased risk. So mild HIN is prevalent and treated, and we've done what we can do in terms of suppressing them in the systemic compartment. How do we treat these people that have ongoing neurocognitive impairment? Do we start, I mean, there is a study that's looking at earlier treatment initiation, and there's certainly early data to suggest the earlier you treat in the disease, the better off the brain is. the longer the virus is in the brain, the worse the brain's going to do. This kind of makes sense. We don't have any novel CNS treatments. We're working on those. We've tried a lot of different things with no success. And we're also looking at intensification strategies. If they're on a stable regimen, they're virally suppressed in the CNS, in the plasma and CSF, maybe we should put them on other drugs that might penetrate better, and we're going to do that study. Here's a study that was presented yesterday by our colleague Bruce Brew in Sydney that looked at a drug called Maraviroc, here abbreviated MVC. It's a small pilot study that went out for a year. It was only in 15 patients, but he had improvement in neurocognitive, and he had an MRS improvement too in glutamate, which I'm not going to show. But in the Maraviroc arm, which is in red here, you can see from baseline, pre-drug, and these are all individuals who had impairment at baseline and were on stable background antiretroviral therapy, and then Maraviroc was added to their regimen. You can see at 6 months and then 12 months that in the Maraviroc arm they got better, and in the placebo arm, or the no additional drug arm, they kind of remain the same. And this, even though it was only a few patients, gave hope for a better study.
Can I ask if there's, do the patients, do they detect a difference in going from 0.8 to whatever it is, 0.5? In other words,
Would that be noticeable to a
Yeah,
patient? In the battery that it sort of depends on how many tests you use as to how big an impact that is. So a half an SD, so if I'm doing a reasonable size battery, which was similar to what they're doing here, and I see half an SD improvement, which is equivalent to this, that's a noticeable clinical change. It's noticeable, I think, to the patient. It's certainly noticeable to me when I see it. So it's a meaningful, people have asked me, well, what do I use for meaningful clinical change across the battery? And a half an SD. So this would be a meaningful clinical change, not just statistical. It's a great question. I hadn't thought about it. Should have thought about it before I presented the slide, but yeah. So we had been working on, we being Christina and a number of others, Serena Spudich at Yale, Scott Latender at UCSF, and Rich Halbert at intensification strategy. So Maraviroc is a R5 inhibitor, and R5 virus is predominant in the CNS, so we think maybe this is a good drug to use to intensify with. There's a new drug that has a novel mechanism, dolutegravir. It's an integrase inhibitor. We think maybe that. We know we have early data that penetrates well into the CNS, and that's a good drug to intensify with. So we have funding to do this study, and it's very far along. Version 1 has just been released to the AIDS Clinical Trials Group sites across the U.S. There are about 30 of those sites. We'll hope 20 or so will participate. Here are the chairs and the vice chairs. Briefly, because I'm running out of time, CNS is a protecting compartment. Some drugs don't get in as well. We hope these will. And we're going to intensify in this manner. These are patients that are virally suppressed on an optimized background drug regimen already, but they have neurocognitive impairment. We're going to have an Arm A with no intensification. They get double placebo. Arm B gets placebo for Maraviroc plus active dolutegravir, and then Arm C gets Maraviroc and dolutegravir active drugs. Not much else to say here. It's 62 per arm, and we're going to stratify by nadir CD4, which seems to be important, and then the severity of their neurocognitive disease, whether it's ANI or MND on entry. To wrap it up, what should we do in the future? Well, I think we need to, because most of the burden of the virus and the disease is in resource-limited settings, to keep building infrastructure there, to try to look at this intensification strategy to solve the neurocognitive problems that we see now, perhaps look at early treatment, and then hopefully we'll come up with novel CNS treatments to address the disease. So thank you very much. I'm happy to take any questions.
Can you talk a little bit more about, so Ravrock's an R5, so it's a CCR5 blocker.
Yes.
And can you talk a little bit about why that might be good for the brain? And can you repeat my question?
So Christina wants me to talk more about why R5 might be good for the brain. There are two types of co-receptors, X4 and R5, and Maraviroc works by inhibiting R5 entry as a co-receptor. And the primary reason is because most of the virus in the brain is R5 typed, according to Sarah Beth, Joseph, and Ron Swanstrom at UNC. And I think there's some evidence that it may have some anti-inflammatory effects as well. Is that what you were getting
Well,
at?
I was thinking about whether it blocks certain types of white cells to get it. So it would block an inflammatory response. So if one of the things that we're thinking about is that there's neuroinflammation because there's low-level virus or some other reason, maybe the way it works is it's not necessarily getting rid of HIV in the brain, but blocking it.
But it has, through its more anti-inflammatory cascade effects. Other questions? Yes.
So I'm curious whether or not you have been able to look at brains that have these different levels of dysfunction, are infected but normal, infected but mild, infected but more severely impaired.
There's been a lot of neuropath done in the past, and there's for the full-blown, even for the full-blown dementia, sometimes there's a disconnect between the neuropathology and the neurocognitive outcomes. But as you get into the more milder forms, there's certainly more disconnect between the neuropathology and the neurocognitive outcomes. There is, on the other hand, data on biomarkers that, like neurofilament light, which is a biomarker of neuronal damage, and early asymptomatic neurocognitive impairment.
I asked that question because if the brain looked essentially normal to modern neuropathology, it would suggest to me that this problem with neurologic function might be completely reversible.
Yes.
If you knew what to do. We know that microglia that are infected release just a panoply of inflammatory molecules, and now we're pulling apart dozens of pathways that are activated, some of them uniquely to astrocytes, back to neurons, and they can be interrupted. There's lots of drugs that interrupt at these different intersections.
It's an excellent comment, and that's what we hope, and you're exactly right, and I'll just repeat the comment and question for the audience because I don't think I did.
They just know it was Bruce and it was about glia.
Well, I mean, the question was, what does neuropathology look like on these early individuals and late individuals, and is your brain already severely damaged with ANI, and thus there's a lack of the ability to recover. And I think I certainly, I'll give you a case example, and it was my first real experience with somebody that had full-blown HIV-associated dementia, shuffling gait, could not talk, mute, had to be led into the clinic by his father, and we, Colin and I went and examined him, and there wasn't much I could do from a neuropsychological standpoint, and Colin gave him a brief neuro exam, pulled the father out, and told him, look, your son's going to die within 3 months, and we're so sorry that you need to start making plans. Joe Eron put him on the first available triple drug regimen, and within 3 months, he had started to talk. Within 6 months, he was arguing with his dad about finances, and then within a year.
He was in medical school.
No, but he did have residual deficits and executive functioning. Was not making. He was still impulsive, but improved dramatically. And so even in those full-blown cases like that, there's enough recovery that can happen that gives us hope for interventions, as you were alluding to.
In your pie diagrams, about half the people do not have any cognitive impairment.
Right.
Are there any clues as to why?
No, but that's a great question. We wish we knew. I mean, I think one of the things that we can correlate with so let me take the converse question, and what correlates with neurocognitive impairment, and that is having the virus longer, having more severe immune dysfunction, so nadir CD4 count, the lowest CD4 count ever, being below 200, so severe immunodysfunction. So if you catch people earlier and suppress the virus, we hope that we'll keep people in the normal category, but it's not proven yet. We're doing a large international trial to look at that called START. So that's what we hope.
I have to say, and it must have occurred to a lot of people listening to you today, Ebola bumped this from maybe not the front page, maybe page 2 to a B section or C section.
Oh yeah.
But Ebola is like a mosquito bite in comparison.
It's scary, man.
It's scary, but this is scary.
This was really scary. I mean, think back to when the early 80s.
Yeah.
Oh yeah.
People wouldn't take care of patients with HIV.
Yeah, and when we went to these sites, I mean, this was no joke. People were dying right and left, and we would go in hospitals, and the infrastructure just wasn't there. I mean, they weren't washing their hands in between patients. The whole 9 yards.
But I mean, there are people being infected like crazy.
Oh yeah, 6,000 a day. Yes. Yes.
The burden of disease, the expensive disease to Ebola, it just completely blots out the horizon, and I'm afraid that this has slipped out of people's minds.
Well, there was I didn't go to this, but there was 2 presentations at the meeting about what Ebola had done to the delivery of AIDS care in places that were hit the hardest. Did you go to those?
No, I did not. No.
But that was an issue that was brought up.
But that's a great point. It really is.
Thank you. That was.
Thank you. Thanks very much for having me. Thank you. Thanks very much.